Good morning everybody. Today I am going to present seminar on gene cloning. I'll be dealing under the following headings. Firstly, introduction. In recent years, techniques for manipulating prokaryotic as well as eukaryotic DNA have witnessed a remarkable development. This has allowed breakage of a DNA molecule at two places to isolate a specific DNA segment and then insert it in another DNA molecule at a right position. The product obtained is called recombinant DNA and the technique is called genetic engineering. Using this technique, we can isolate and clone single copy of a gene or a DNA segment into an indefinite number of identical copies. This is possible because bacteria, phages and plasmids reproduce in their usual manner even after insertion of a foreign DNA. So that inserted DNA will also replicate faithfully with the parent DNA. This technique is called gene cloning. Now coming to the definition proper. Isolating a gene and joining it to a vector DNA called recombinant DNA molecule, which are then propagated in a host cell which produces a clone that contains a single fragment of the target DNA. Clone is a set of absolutely identical molecules, cells or even organisms produced by asexual propagation. A century ago Gregor Mendel made a basic assumption that each heritable property of an organism is controlled by a factor called gene. In 1900, Mendel's law was introduced, the birth of genetics. What these genes are and how exactly they work were spoken about. In 1903, Sutton W. proposed that genes reside on chromosomes. In 1910, Morgan Th. made an experimental backing on that, telling that development of the techniques for gene mapping, that is to establish the structures or structural details or location. By 1922, a comprehensive analysis of the relative position of over 2000 genes on the 4 chromosomes of the fruit fly, also called as Drosophila melanogaster. In 1952, Hershey and Chase did experimental results were shown that DNA is the genetic material. Conventional idea was made that genes were made of proteins. In 1952 to 1966, Delbruck, Shargaff, Crick and Monod, the structure of DNA, genetic code and the process of transcription and translation were described. In 1971 to 1973, Stanley Cohen, Herbert Boyer and Paul Berg, new experimental techniques were developed by them and recombinant DNA technology or genetic engineering based on the process of gene cloning was introduced. This led to rapid and efficient DNA sequencing techniques that enabled the structures of individual genes to be determined. In the 1990s, it started with massive genome sequencing projects, including the Human Project. This led to procedures for studying the regulation of individual genes. Joshua Lederberg introduced the term plasmid. Genome, a complete single set of a genetic material of a cell or an organism. From the techniques, modern biotechnology is appeared for the production of proteins and other compounds needed in medicine and industrial process. Why gene cloning is so important? Gene isolation by cloning. Cloning can provide a pure sample of an individual gene. The problem of selection of just one gene is overcome. By cloning, only cells containing the desired recombinant DNA molecule can divide and the clone of interest is automatically selected. Coming to the basic steps of gene cloning. The first step is when a fragment of DNA containing the gene to be cloned is inserted into a circular DNA molecule, which is also called as vector, a recombinant DNA molecule or a chimera is formed. The vector acts as a vehicle that transports the gene into a host cell, usually a bacteria, possibly other types of living cell. Within the host cell, the vector multiplies, producing numerous identical copies not only of itself but also of the gene that it carries. When the host cell divides, the copies of recombinant DNA molecule are passed to the progeny and the further vector replication takes place. After a large number of cell division, a colony or clone of identical host cells is produced. Each cell in the clone contains one or more copies of the recombinant DNA molecule. The gene is cloned. Coming to cloning vehicles and clone. Cloning of DNA is possible only with the help of another DNA molecule which is capable of replicating in the host. This other DNA molecule is used in the form of a vector. A wide variety of different cloning vectors are available in the market. All are derived from naturally occurring plasmids, bacteriophages, or viruses. Those could be modified in various ways. Coming to cloning vehicles. DNA molecule must be capable of entering a host cell and replicating to produce multiple copies of itself without disrupting the host cell functions. Properties of a vector. This can independently replicate, the vectors can independently replicate themselves and the foreign DNA segments they carry. They contain a number of unique restriction endonuclease cleavage sites that are present only once in the vector. They carry a selectable marker to distinguish host cells that carry vectors from host cells that do not contain a vector, usually in the form of antibiotic resistance genes or genes for enzymes missing in the host cell. The vectors are easy to recover from the host cell. Plasmids. These are autonomous elements whose genomes exist in the cell as extra chromosomal units. These are a small circle of DNA found in bacteria and other organisms. Plasmids can replicate independently of the host cell chromosome. There are two types of plasmid, that is a single copy plasmid and multiple copy plasmids. This is a picture showing a plasmid which confers resistance to ampicillin and tetracycline. A foreign DNA fragment is this plasmid undergoes ligation by DNA restriction endonuclease. The cut part is filled by foreign DNA fragment, forming the required vector. Selection of DNA is facilitated by resistance genes which the plasmid carry against a certain antibiotic. Sometimes, if two resistance genes are present, the DNA selection becomes difficult and it has to be restructured by inserting genes of relaxed replication. Some of the examples are PBR322 and PUC vectors. This PBR322 is obtained from E. coli 4362 BODNA and derived by several alterations. Three plasmid vectors used for gene cloning. The structure of PBR322, it is a vector used with E. coli. The structure of PHV33, it is a vector which can replicate in both E. coli and B. subtilis. The structure of YRP17, a vector which can replicate in both E. coli and yeast. Coming to conjugation and compatibility. Conjugative plasmids. The ability to promote sexual conjugation between bacterial cells. Conjugation and plasmid transfer are controlled by transfer of a set of genes. Non-conjugative plasmids. Absence of these genes and under some circumstances it may be co-transferred along with a conjugative plasmid when both are present in the same cell. Plasmid classification. It is based on the main characteristic coded by the plasmid genes. Firstly, fertility or F plasmid. They promote conjugal transfer of plasmids. Example, F plasmid of E. coli. Resistance or R plasmids. They carry genes conferring on the host bacterium resistance to one or more antibacterial agents. Example, RP4 in Pseudomonas. Col plasmids. They code for colicins, the proteins that kill other bacteria. Example, Col E1 of E. coli. Degradative plasmids. This allows the host bacteria to metabolize unusual molecules such as toluene and salicylic acid. Example, TOL of Pseudomonas putida. Virulence plasmids. They confer pathogenicity on the host bacterium. Example, TI plasmids of Agrobacterium tumefaciens. Coming to plasmid purification. A culture of bacteria is grown and then harvested. The cells are broke open to release their contents. This cell extract is treated to remove all components except the DNA. Purification of plasmid DNA from total cell DNA is on the basis of size and conformation. This is a detailed view which shows the chemical lysis by cell disruption. Here, the bacterial, we can see a host cell with a bacterial chromosome and many plasmids. This undergoes lysis by lysozyme and EDTA with 25% sucrose. What lysozyme does is it digests the polymeric compounds that give the cell wall its rigidity. EDTA removes magnesium ions that are essential for preserving the structure of the cell and also inhibits cellular enzymes that could degrade DNA. Here we can see the intact cell membrane and also the disrupted cell wall. This is again treated with detergent like Triton X100. This detergent will aid the process of lysing by removing the lipid molecules in the cell membrane. Therefore, the cell extract is now outside. This cell extract is again centrifuged. Upon centrifuging, we get the cell debris which is in the bottom, and large DNA fragments, and on the top layer will be a cleared lysate containing plasmids. Bacteriophages. A phage has a linear DNA molecule. A single break will generate two fragments which are joined together with foreign DNA to generate a chimeric phage particle. Examples EMBL3 and 4. They are also known as replacement vectors. A central non-essential part can be replaced by a foreign DNA. Caron 34 and 35. M13. These are filamentous bacteriophage of E. coli and they contain 7.2 kilobase long single stranded circular DNA. These are used for Sanger's method of sequencing. Plasmids are plasmid particles into which certain specific DNA sequences are inserted. They perpetuate into bacteria and do not carry genes for lytic development. Here is a picture showing the gene transfer in bacteria. This is a bacteria with a plasmid containing the DNA. Now this is removed as a vector. This plasmid is being used as a vector. Into this vector, a foreign gene is inserted. This will become a recombinant DNA molecule after the insertion of foreign gene. Now this plasmid or vector is transferred into a host cell, and this host cell divides and all the daughter cells contain the same vector. Gene transfer in bacteria. Transformation. A bacteria takes up DNA from the medium. Recombination takes place between introduced genes and the bacterial chromosome. Methods of transformation are electroporation, freeze-thaw method, calcium chloride mediated transformation, calcium phosphate mediated transformation. The most common method used is calcium chloride method. Calcium chloride method. Here, treatment with calcium chloride in the early log phase for growth for competence. Bacterial cell membrane is permeable to chloride ion but is non-permeable to calcium ion. As the chloride ions enter the cell, water molecules accompany the charged particle. Here we can see influx of water causes the cell to swell and is necessary for the uptake of DNA. Here we can see in this picture the DNA of interest is added into the cell. Calcium chloride treatment is followed by addition of DNA of interest, then by heating. The heat shock step is necessary for the uptake of DNA. All the cells recover and the plasmid genes are expressed. The bacterial colonies are selected using antibiotic selection techniques. Here in this picture we can see a schematic diagram. First, this is a starter plate into which ampicillin sensitive E. coli cells in log phase of growth are transferred into a beaker containing cold calcium chloride solution. From this, the ampicillin resistant plasmids are added to experimental cells only. Now this is incubated or shocked at 42 degrees Celsius, that is the heat shock step. Some of the competent cells take up the ampicillin resistant plasmid and they are transformed. The treated cells are spread on an agar plate containing ampicillin. The ampicillin kills the cells that lack ampicillin resistant gene. Now this cell is again incubated for this agar plate is incubated for 24 hours in 37 degrees Celsius. And now we obtain a agar plate which contains only colonies of E. coli that have been transformed by the ampicillin resistant gene. Now coming to gene transfer in virus. The viral DNA acts as a vector. Into the viral DNA, a foreign gene is inserted and the recombinant DNA molecule is formed. Now this is introduced into a host cell, and here the DNA in the host cell multiplies, and then the cell lysis occurs. When we then we obtain many of these viruses containing the vector. Virus chromosomes. A number of viruses are used as vectors for both introducing foreign DNA into cells and for gene amplification. The chromosomes of bacteriophages, that is viruses that specifically infect bacteria. During infection, the bacteriophage DNA molecule is injected into the host cell where it undergoes replication. Some of the viruses used are adenovirus, papovavirus and retroviruses. Some other known vectors are transposons, artificial chromosome vectors for cloning large DNA segments. Used are plasmid, which clones up to 10 to 15 kilobases, lambda phage, that is also bacteriophage, it clones up to 22 kilobase units, cosmid up to 40 kilobases, yeast artificial chromosome vector, this clones several hundred kilobase per long vectors. Now the problem of selection. Even the simplest organisms like E. coli contain several thousand genes, and a restriction digest of total cell DNA will produce not only the fragment carrying the desired gene but also many other fragments carrying all the genes. During the ligation reaction, there is of course no selection for an individual fragment. Numerous different recombinant DNA molecules are produced, all containing different pieces of DNA. A variety of recombinant clones are obtained after transformation and plating out. After that, the correct one must be identified. Now coming to restriction enzymes used for cloning. For cloning of DNA, we need to cut the DNA at specific sites, which are recognized and cleaved by specific enzymes. Those are endonucleases, which cleave DNA at interval sites. These are described as restriction enzymes. These enzymes cleave at specific nucleotide sequences called recognition sites. These enzymes recognize short sequences of double-stranded DNA as targets for cleavage. These enzymes are specific for each sequence and they are highly specific deoxyribonucleases. Each enzyme is named by a 3-letter abbreviation that identifies its origin. Roman numerals such as 1, 2, etc. are added to distinguish several enzymes within the same origin. For example, EcoRI, R1 derived from E. coli and HPAI from Haemophilus parainfluenza. Restriction maps are prepared by locating the position of cleavage sites by these enzymes. Besides cleavage, modification in the form of methylation is also brought about by enzymes called modification enzymes, also known as methylases. Restriction enzymes are grouped into they are grouped into 4 classes. The most common ones are type 1, 2 and 3. Type 2 restriction enzyme system, example are EcoR1, they separate enzymes for modification and restriction. Type 1 and type 3 are bifunctional enzymes. Here we can see the 4 types of enzymes: type 1, type 2, type 3 and type 2S. Type 1, this is a one enzyme with different subunits for recognition, cleavage as well as methylation. They recognize and methylates a single sequence but cleaves DNA up to 1000 base pair away. Type 2, two different enzymes which both recognize the same target sequence, which is symmetrical. The two enzymes either cleave or modify the recognition sequence. Type 3, one enzymes with two different subunits, one for recognition and modification and one for cleavage. They recognize and methylase same sequence but leaves 24 to 26 base pair away. Type 2S, these are two different enzymes but recognition sequence is asymmetric. Cleavage occurs on one side of recognition sequence up to 20 base pair away. This is the example of the nomenclature of few of the enzymes. The EcoR1 is derived from Escherichia coli and its recognition site is at 5-GAA here and 3-CTT here. So it cleaves the DNA in this area. Similar examples are HPAA1, 2 derived from Haemophilus parainfluenza, MSP1 derived from Moraxella species, and PUV1 which is derived from Proteus vulgaris. Sticky end. Some enzymes cause the two helicases a few base pair apart, generating two fragments with single strand protrusions called sticky end. Blunt end or flush end. Some enzymes make a simple double-stranded cut in the middle of the recognition sequence. Here we can see how the recognition enzyme acts. The recognition enzyme cut covalent phosphodiester bonds of both strands. This is a normal DNA. The restriction enzyme, they cut the covalent phosphodiester bonds of both the strands, often in a staggered way, creating single-stranded end. Here we can see this, the end is a single-stranded end. These are called as sticky ends. To this sticky end, DNA fragment added from another molecule cut by the same enzyme is added, and then base pairing occurs. These extensions will form hydrogen base pairs with the newly inserted DNA, and the longer DNA is formed. The DNA ligase enzyme, it will seal these gaps. The bonds which are caused by the restriction enzyme, the DNA ligase enzyme will seal that bond, forming it a continuous DNA molecule. The extracted genomic DNA is digested with the restriction enzyme and cleaved into segments which can be separated into different sizes. The techniques used are separation of DNA fragments using agarose or polyacrylamide gel electrophoresis, separation of large DNA molecules using pulsed field gel electrophoresis, and contour clamped homogeneous electric field electrophoresis. Southern, northern and western blotting, dot blots and slot blots. Two basic strategies for obtaining the correct clone are direct selection and clone identification. In direct selection, plating out method is used. It is only the correct DNA, recombinant DNA can survive in this method. The simplest examples of direct selection occurs when the desired gene specifies resistance to an antibiotic, example kanamycin or ampicillin. Identification of the clone from a gene library. Analysis of the individual clones to identify the correct one. Problems of marker rescue are many bacterial mutants are not auxotrophs. Foreign genes from animals and plants sometimes do not function in the bacterial cell. Coming to the applications of gene cloning, a particular gene can be isolated and its nucleotide sequence can be determined. They control the sequences of DNA can be identified and analyzed. Protein, enzyme or RNA function can be investigated. Mutations can be identified using gene cloning. Example, gene defects related to specific diseases. Organisms can be engineered for specific purposes. Example, insulin production, insect resistance, etc. Gene cloning is also used a lot in horticulture and plant department. Here is a picture showing the summary of gene cloning. This is the host bacteria. This is the host bacteria which contains the bacterial chromosome as well as the plasmid. The plasmid is removed from it. This is the cell of interest from which the DNA is isolated. The gene of interest is added to the plasmid which forms a recombinant DNA or a vector. This plasmid is inserted into a bacterial cell. So there is a recombinant bacteria. This bacteria will become a recombinant bacteria. They will replicate. The host cell grown in culture to form a clone of cell containing the clone gene of interest. These are all the replicated host cells. From this host cell, the protein expressed from the gene of interest is isolated. The protein is harvested and it is used for basic research on protein. like it can be used for human growth hormone production, which will treat the stunted growth, and also protein which dissolves blood clots in heart attack therapy, like streptokinase. Then there is the another use of gene of interest, which can be made into multiple copies, and they can be used for pest resistance pest resistance genes, which are inserted into the plants, and also they are used to alter bacteria for cleaning up toxic waste. Now coming to the gene library. A genomic library is made by collection of recombinant vector clones produced by cloning DNA fragments from an entire genome. A clone carrying the gene of interest can be identified with a nucleic acid probe having a sequence complementary to the gene. This process is called nucleic acid hybridization. A probe can be synthesized that is complementary to the gene of interest. The DNA probe can be used to screen a large number of clones simultaneously for the gene of interest. Once identified, the clone carrying the gene of interest can be cultured. Now coming to a short note on PCR cloning method. PCR cloning differs from traditional cloning in that the DNA fragment of interest and even the vector can be amplified by the PCR and ligated together without the use of restriction enzyme. PCR cloning is a rapid method for cloning genes and is used for projects that require higher throughput than traditional cloning methods can accommodate. It allows for the cloning of DNA fragments that are not available in large amounts. Typically, a PCR reaction is performed to amplify the sequence of interest, and then it is joined to the vector via a blunt or single base overhang ligation prior to transformation. Early PCR cloning often used TAQ DNA polymerase to amplify the gene. This results in a PCR product with a single template independent base addition of an adenine residue to the 3' end of the PCR product through the normal action of the polymerase. These A-tailed products are then ligated to a complementary T-tailed vector using T4 DNA ligase followed by transformation. High fidelity DNA polymerases are also routinely used to amplify sequences with a PCR product containing no 3' extensions. The blunt end fragments are joined to a plasmid vector through a typical ligation reaction or by the action of an activated vector that contains a covalently attached enzyme, typically topoisomerase 1, which facilitates the vector, that is the insert joining. Some PCR cloning systems contain engineered suicide vectors that include a toxic gene into which the PCR product must be successfully ligated to allow propagation of the strain that takes up the recombinant molecule during transformation. A typical drawback common to many PCR cloning methods is a dedicated vector that must be used. These vectors are typically sold by suppliers like NEB in a ready-to-use linearized format and can add significant expense to the total cost of cloning. Also, the use of specific vectors restrict the researcher's choice of antibiotic resistance, promoter identity, fusion partners, and other regulatory elements. Here is a table summarizing gene cloning and PCR. In gene cloning, a gene amplification technique where recombinant DNA is constructed in vitro and is amplified in vivo inside a bacteria. In PCR, a gene amplification technique where the DNA is amplified in vitro, no need for construction of recombinant DNA. Restriction enzymes, DNA ligase, vector DNA and bacterial cells are all required. In PCR, TAQ DNA polymerase or a thermostable DNA polymerase, RNA primers and free deoxyribonucleotides are required along with DNA segment to be amplified. In gene cloning, at least a microgram quantity of DNA is required for amplification, whereas in PCR, a nanogram of DNA is enough for amplification. In gene cloning, a restriction enzyme is required for reisolation of the amplified DNA from recombinant DNA. In PCR, there is no need for reisolation or use of enzymes. Gene cloning requires 2 to 4 days for the experiment to be processed, whereas PCR requires a minimum of 4 hours for the complete experiment. There is no need of any automation in gene cloning, whereas automation is a must in PCR. Gene cloning is labor intensive, whereas PCR does not need much of intensive labors. Gene cloning error possibility is more. In PCR, error possibility is less. Gene cloning can be preferred over PCR only because a dedicated vector can be used in gene cloning, which cannot be used in PCR. These are my references. Thank you.